Yeah, hi, good evening. So today I'll start the fifth problem solving session for the course Overview and Integration of Cellular Metabolism. So the course instructors, as you know, are Professor Ritribir and Professor Indam Ghosh. They're from IIT Kharagpur, and then I'm Shrija Ghosh, a PMRF from IIT Kanpur. I'm here to help you solve the assignment questions that are given in this week's session. So this week is all about lipoprotein metabolism and fatty acid catabolism, that is fatty acid breakdown or fatty acid oxidation. Okay, so we'll discuss different questions related to these two topics, and feel free to ask any question or any doubt if you have in any of the topics that are discussed. Uh, if you have any other doubt which are not discussed in today's session, let me know. Like if I have missed out any topic from week five's lecture, let me know. I'll try to explain that. So with that, I think I can start my class. Okay. So starting with the first question. Activated lecithin cholesterol acyl transferase is essential for the conversion of option A VLDL remnants to into LDL, option B nascent HDL into HDL, option C HDL2 to HDL3, and option D HDL3 into HDL2. Activated lecithin cholesterol acyl transferase is essential for the conversion of VLDL remnants into LDL, nascent HDL into HDL, HDL2 into HDL3, and HDL3 into HDL2. If you know the answer, you can write it in the chat box. Option B, that is nascent HDL into HDL, that is the correct answer. So, less than cholesterol acyl transferase, it's a protein, it's an enzyme. Basically, it transfers uh, it transfers the ester group from lecithin into the cholesterol. So basically cholesterol becomes cholesterol ester. So it's a part of HDL metabolic pathway. So this enzyme LCAT, it's the short form is LCAT. It is associated with the HDL lipoprotein, and it esterifies the cholesterol and adds to disc shaped nascent HDL to mature spherical HDL. So basically, nascent HDL converts into mature HDL by the help of this enzyme LCAT. It converts cholesterol and phosphatidylcholine of chylomicron and VLDL remnants into cholesterol ester, and there are different apoproteins that are associated with the HDL, which stimulates this LCAT activity. So basically, this lecithin molecule, the fatty acid or the ester group, this is transferred to the cholesterol. So it becomes cholesterol ester. Okay, so this is the HDL metabolic pathway where so there are two locations. One is liver and one is the extrahepatic tissue, basically all the other tissues other than liver. Okay, now in the liver the cholesterol synthesis will happen, and HDL metabolisms actually start, like HDL uh, formation actually starts from a molecule called pre-beta HDL. So this pre-beta HDL has the apoprotein A1 associated with it, and it mostly consists of phospholipids and cholesterols. Okay, now this pre-beta HDL will become a nascent form of HDL, that is discoidal HDL, uh, in which LCAT will associate, and that LCAT will now convert all the cholesterol into cholesterol ester. So here you see phospholipid and cholesterol. This cholesterol is now here converted to cholesterol ester, and some cholesterols are still there. So all the cholesterol will slowly get converted into cholesterol ester and then phospholipid. This form of HDL is called HDL3. Now then further this HDL3 gets converted to HDL2 by another protein, which we'll discuss in detail in the further questions, and later the cholesterol is then again transferred to the liver. So basically this pre-beta HDL, it takes up all the cholesterol from the extrahepatic tissues, that is all the tissues that are outside the liver, and it transports the cholesterol back to the liver. So it's kind of a cycle, cholesterol cycle. Also HDL is important because it's a carrier of all these other apoproteins that are required for VLDL and LDL uh, metabolism. Okay, so LCAT or lecithin cholesterol acyltransferase, it actually converts the cholesterol to cholesterol esters. Okay. Okay. This is clear. Moving on to the next question. Which of the following is an integral apolipoprotein uh, present in a chylomicron? Which of the following is an integral apolipoprotein present in chylomicron? Option A, ApoB100, option B, ApoB48, option C, ApoC2, and option D, ApoE. So these are different apoproteins. So which of the apoprotein is the integral apolipoprotein or apoprotein in a chylomicron? You're saying option B, that is ApoB48. That is the correct answer. So ApoB48 is the integral apolipoprotein. So all the lipoproteins have a integral uh, protein. So it's by the name itself you understand, like lipoproteins are they comprise of lipids and proteins. So all the lipoproteins, they have some integral apolipoproteins or apoproteins and some peripheral apoproteins. Okay, the integral ones are always present, they are like the markers. So if ApoB48 is present, that means that particular lipoprotein is chylomicron. Okay, so it's like a marker. So this is the chylomicron. So this is metabolism of metabolic pathway of chylomicron. So in the previous class, if you remember, we have discussed that chylomicrons are the uh, lipoproteins that carry the triacylglycerol from the diet, uh, from the like all the dietary triacylglycerol is absorbed by this chylomicron, which helps in the transport of these uh, fat lipids or these cholesterols into different locations. So chylomicron has high amount of triacylglycerol and some amount of cholesterol, and ApoB48 is the integral protein, so that is like the marker of uh, chylomicron. Along with that, it also has this ApoA protein associated. Then this is the nascent form of chylomicron. Then when it matures, this is the mature form of chylomicron. So the mature form, yeah. So the mature form has ApoC and ApoE protein. So additional two more apoproteins are there. So B48 is integral, it is always present. ApoA is present in the nascent one, and then from that it becomes mature when ApoE and ApoC adds to it. Okay, now this is the mature chylomicron. From this, these triacylglycerol will keep on metabolized by these lipoprotein lipase, and the fatty acids will be absorbed by the extrahepatic tissues. The glycerol will be going into the metabolic pathways, and this chylomicron will keep shrinking in size, so it will become smaller and smaller because now the triacylglycerol content is uh, getting lowered. So cholesterol, obviously the ratio of cholesterol to triacylglycerol will increase, and then this chylomicron remnant that is there, which has high cholesterol and low triacylglycerol, that is transferred to the liver by two different receptors, and uh, also this chylomicron is transferred is converted into HDL, so that we'll discuss later. So this is the chylomicron metabolic pathway. So B48 is basically a integral lipoprotein for chylomicron. It's like a marker. Okay, clear? Okay, moving on to the next question. The highest phospholipids content is found in. The highest phospholipid content is found in option A chylomicrons, option B VLDL, option C LDL, and option D HDL. Highest phospholipid content is found in chylomicron, VLDL, LDL, and HDL. You are saying option D, that is HDL. That is the correct answer. So chylomicrons, as I mentioned, it has the highest amount of triacylglycerols. Uh, then comes the VLDL, which is very low density lipid, then low density lipid, and high density lipid. So as we so chylomicrons are the largest particle and they have the least density, and as the density increases the size decreases, and HDL is a small particle with high density. Okay, and it has the highest amount of phospholipids present. So this is kind of a overview of the whole lipoprotein metabolism. So this is the HDL. HDL molecules are basically carriers of phospholipids and cholesterol along with certain apoproteins. So all these apoproteins that are required for the metabolism of different lipoproteins, they all like most of them are carried by this HDL. Okay, so ApoC2, ApoA, ApoE, all of these are carried. So we saw in nascent chylomicron, for the maturation of chylomicron from nascent chylomicron, we need ApoE and ApoC2, that is given by this HDL. Also for VLDL, same. So like chylomicron has ApoB48 as the integral apoprotein. Nascent VLDL has ApoB100 as the integral apoprotein. Other than that, its maturation is also dependent on ApoE and ApoC2, which comes from HDL. Okay, and then HDL, I told you, it absorbs all the cholesterol from the extrahepatic tissue outside the liver, and then later that cholesterol is converted to cholesterol ester by LCAT, and this cholesterol ester actually is transferred, like exchanged in the VLDL and LDL, and glycerols are transported from VLDL to HDL. So initially HDL does not have enough triacylglycerol, but later at a later stage, the mature form of HDL has triacylglycerol, and the cholesterol esters are transported out of the HDL to these VLDLs, and then these VLDL can become IDL and LDL, and then slowly these cholesterol can be metabolized by the liver, so it can reach the liver where it can have multiple feeds. Okay, so this is a compiled kind of lipoprotein metabolism overview. This is clear? Any doubt in this? Okay, moving on to the next question then. Familial hypercholesterolemia is a genetic disorder of cholesterol metabolism. The defect lies in the familial hypercholesterolemia is a genetic disorder of cholesterol metabolism. The defect lies in the dash. Option A, transport of cholesterol from extrahepatic tissue to the liver. Option B, impairment of cholesterol degradative pathway. Option C, impairment of uptake of cholesterol by tissues. And option D, impairment of HDL metabolism due to deficiency of ApoA. Familial hypercholesterolemia is a genetic disorder of cholesterol metabolism, and the defect lies in the option A, transport of cholesterol from extrahepatic tissue to liver. Option B, impairment of cholesterol degradative pathway. Option C, impairment of uptake of cholesterol by the tissues. And option D, impairment of HDL metabolism due to deficiency of ApoA. What is the correct answer here? Do you know the correct answer? Even if you don't know, you can guess. You are saying transport of cholesterol from extrahepatic tissue to the liver. The correct answer is actually impairment of the uptake of cholesterol by the tissues. So it's kind of both the options are similar, but then the tissues have a mechanism to uptake the cholesterol, uh, not from the liver, or it is actually base, so here it is. So basically LDL. So, familial hypercholesterolemia is a defect in the LDL receptors, okay, or mutation in the ligand region of ApoB100 that binds to the LDL receptors. So, this LDL receptors are mainly responsible for transport of the cholesterol from the liver to the extrahepatic tissue. So your option A was from extrahepatic tissue to the liver. So it's the opposite actually. Uh, so you see, this is the LDL, VLDL metabolism. So VLDL has ApoB100 as the integral protein, and the mature form has the ApoE and ApoC2. Then this VLDL, once matured, it will basically by the lipoprotein lipase, the triacylglycerol will keep converting into fatty acids, the glycerols, and when it becomes smaller and smaller, it's called the VLDL remnant or the IDL. It will, after some point, all the triacylglycerol will be metabolized, and only thing that is remaining is cholesterol. In that case, that is called the LDL, or low density lipoprotein. So that LDL can either go into the liver by the LDL receptor present in the liver, that is the hepatic LDL receptor, and there the cholesterol is taken up by the liver. And also LDL can enter the extrahepatic tissue by the same receptor, where it can transport the cholesterol into the liver. So basically, due to the defect in the LDL receptor, it's mainly the defect in the LDL receptor of the extrahepatic tissue. There are elevated levels of LDL, and it leads to hypercholesterolemia. It results in atherosclerosis and coronary disease. Okay, so LDL, which originates from the VLDL remnant, they contain endogenous cholesterol and phospholipids with minimal triacylglycerol. So that I told that when all the triacylglycerols are metabolized, only mainly cholesterol and some amount of phospholipids are present in LDL, and it also contains that ApoB100 because it is coming from VLDL only, and VLDL has this ApoB100 as integral lipoprotein. Then it functions in transporting endogenously biosynthesized cholesterol from liver to the peripheral or extrahepatic tissue, as I mentioned. So this is the LDL receptor, how it works. So basically, the LDL receptor has the affinity towards this ApoB100 protein. Okay, so ApoB100 induced identification. The LDL receptor will identify the ApoB100 and let it bind to it. Once this LDL particle binds to the LDL receptor, the uptake will be receptor mediated endocytosis. So LDL receptor complex formation happens, complexes of LDL and receptor internalization by receptor mediated endocytosis. Then once after the endocytosis, lysosome will fuse to the endosome to form endolysosome. Before that, you see this receptor is recycled back to the membrane so that more and more LDL particles can bind. And then this endolysosome, basically inside this endolysosome, all the lysosomal lipases and proteases will be present, and they will degrade the LDL. Cholesterol is then incorporated into the cell membrane or is stored as cholesterol esters, and ApoB100 is degraded to amino acids. So basically, this is how cholesterols are transported to the cell membranes, or cholesterol are stored in our body for later to be used for energy. Okay, so this is so in the familial hypercholesterolemia, basically this receptor is defective, or there's mutation in the ApoB100 protein, so this binding does not occur. Once this binding does not occur, LDL will keep accumulating here. There won't be any internalization or metabolism of cholesterol. Okay, that's clear. Is that clear? Okay, so coming to the next question, which of the following best describes the role of cholesterol ester transfer protein, CETP? Okay, which of the following best describes the role of cholesterol ester transfer protein, CETP? Option A, it converts cholesterol from LDL to cholesterol esters. It is option B, it is the major protein of HDL. Option C, it exchanges cholesterol esters from HDL to other lipoproteins. Option D, it acts as a ligand for hepatic receptors of HDL. Option E, it is activator of LCAT, that is the less than cholesterol acyl transferase. Okay, so which of the following best describes the role of cholesterol ester transfer protein, CETP? Converts the cholesterol from LDL to cholesterol esters, or it is a major protein of HDL, or is it uh, uh, exchange it exchanges the cholesterol esters from HDL to lipoproteins to other lipoproteins? Then it acts whether it acts as a ligand for hepatic receptors of HDL, or whether it acts as an activator of LCAT. What do you think is the answer? You can write the answer in the chat box. You're saying option A, that it converts the cholesterol from LDL to cholesterol esters. Do you remember the role of LCAT? We discussed the role of LCAT, right? Yeah, what was it doing in HDL? What was it doing in HDL? LCAT? You can unmute and answer, it's okay. Nascent HDL conversion. And how was it doing the nascent HDL conversion? With what molecule was actually changing there? What molecule was actually changing there? How was it becoming nascent HDL? Like how was it becoming mature HDL from nascent HDL? We just discussed this. That was the first question we discussed. Do you remember? It's okay, even if you don't remember, I can re- like revisit the whole thing. Are you confused in this part? Okay, I'll explain. So basically, I think I'll open the HDL metabolism first. Huh. So here, so basically, this is the pre-beta HDL, and this pre-beta HDL gets converted into discoidal form of HDL, and later it matures to HDL. So this discoidal form of HDL is the nascent HDL. Okay. Now, if you see in this HDL, we have cholesterol, and there's an enzyme called LCAT that is converting this cholesterol into cholesterol ester. So remember, I've shown you this. So this is lecithin. Lecithin has this fatty acid chain which gets converted to this OH group of the cholesterol by an ester bond. So now this cholesterol has become cholesterol ester. Okay. So this cholesterol ester can then further be transported and metabolized. Okay, whatever. So LCAT is basically doing the conversion of cholesterol to cholesterol ester in the HDL so that the nascent HDL can become mature HDL. So when it has cholesterol, it is nascent HDL. When it becomes cholesterol ester, it is the mature HDL. Okay. So this part is clear. LCAT is the enzyme that converts cholesterol to cholesterol ester, making the nascent HDL to mature HDL. Now we have the cholesterol, some cholesterol which is still getting converted to cholesterol ester. Then we have the cholesterol ester and phospholipid and HDL. And this form of HDL is called HDL3. Now there's another enzyme called CETP which exchanges these cholesterol esters to different other lipoproteins. So now I'll come back to the question. So it basically, the answer to this question is it exchanges the cholesterol ester from HDL to other lipoproteins. So once the HDL3 has enough cholesterol esters, now that cholesterol ester will be exchanged basically in exchange of triacylglycerol. So HDL3 will give the cholesterol esters to LDL and VLDL, and in return VLDL and LDL, they will give the triacyl glycerols to this HDL, and the HDL with more triacyl glycerol and less cholesterol ester, it is called the HDL2. So basically, CETP, that is the cholesterol ester transfer protein. Now it's a transfer protein, okay? So it has to transfer something. What is it transferring? It is transferring cholesterol esters. Where it is transferring? It is giving it to the other LDL and VLDLs, okay? Other lipoproteins. And in exchange, what it is taking? It is taking triacyl glycerols. So you see, presence of triacyl glycerol increases the size. It's a low density, it decreases the density and increases the size of the lipoprotein. Okay, so HDL3 is a smaller lipoprotein which has cholesterol esters. Now these cholesterol esters are exchanged by CETP. So CETP, or C transfer protein, cholesterol ester transfer protein, it will transfer the cholesterol and acquire the triacyl glycerol, make converting the HDL3 into HDL2. HDL2 is slightly bigger in size because it has more triacyl glycerol and lesser cholesterol esters. Where the cholesterol esters went to the other lipoprotein, that is LDL and VLDL. So CETP, or cholesterol ester transfer protein, it is responsible for the formation of subtractions of HDL. HDL2 is converted to HDL3, and prior to CETP activity, HDL is smaller particle termed as HDL3. I told you it is CE rich, so it is smaller. Post CETP activity, HDL3 becomes larger, TAG rich, uh, TAG rich, and it is termed as HDL2 because it has both triacylglycerol and some amount of cholesterol esters left. So CETP and LCAT. So LCAT will keep converting the cholesterols present in the pre-beta HDL into uh, cholesterol esters. These cholesterol esters will be transferred to LDL and VLDL in exchange of transacyl glycerols, which will be in the final HDL2 particle. Okay. CETP can also transfer these cholesterol esters back to the pre-beta HDLs. Okay. Is this clear now? Okay, good. So the question was, which of the following best describes the role of cholesterol ester transfer protein? So it exchanges the cholesterol ester from HDL to other lipoprotein. Okay. Moving to the next question now. Where are the enzymes for beta oxidation present? What is the site of beta oxidation basically? Where are the enzymes of beta oxidation present? You're saying option C, that is mitochondria. That is the correct answer. So, beta oxidation occurs in mitochondria. So, if you remember from the previous sessions, what was the other uh, cycle that was occurring in mitochondria? What was the other cycle that was happening in that was occurring in mitochondria? TCA, yes. So, and do you know the product of beta oxidation? Major product of beta oxidation of fats? Acetyl coenzyme. So basically, acetyl coenzyme is the substrate for TCA, one of the substrate for TCA. So this acetyl coenzyme in the mitochondria on the beta oxidation is happening. More and more acetyl coenzyme will keep forming, and that acetyl coenzyme will enter into another cycle that is happening in mitochondria, that is TCA. Okay. And along with that, during beta oxidation, we'll see in the upcoming slides, we have NADH and FADH2 formation. So those NADH and FADH2 can also enter the electron transport chain which is responsible for the ATP production, and that also occurs in the mitochondrial membrane. So, uh, like nature has planned the things in such a way that everything is tightly regulated. Okay. The complete oxidation of odd chain fatty acid produces which of the following? Complete oxidation of odd chain fatty acid produces which of the following? Option A, acetyl coenzyme A only. Option B, acetyl coenzyme A and propionyl coenzyme A. Option C, butyryl coenzyme A. Option D, palmitoyl coenzyme A. Option E, propionyl coenzyme A only. Complete oxidation of odd chain fatty acid produces which of the following? Acetyl coenzyme A only. Option B, acetyl coenzyme A and propionyl coenzyme A. Option C, butyryl coenzyme A. Option D, palmitoyl coenzyme A. And option E, propionyl coenzyme A only. What is the correct answer? You are saying option B. That is acetyl coenzyme and propionyl coenzyme. That is the correct answer. So basically, in beta oxidation, whatever the carbon number is, so let's say it is 16. So after every cycle of beta oxidation, 2 carbons will be less because we are having one acetyl coenzyme A remove after every cycle. So acetyl coenzyme A is a uh, two carbon compound. Okay. Now, if we have an odd chain fatty acid, that is, let's say 15 or 17. So once two carbons are removed, how many will be there? C13, or in this C15. Then again 2 will be removed, then it will be C11, and if this cycle keeps on continuing, you will get C5. After some point you will get C5, and from this, if I now remove 2, you will get C3 plus C2. So C2 is basically acetyl coenzyme A, and C3, the 3 carbon, is called the propionyl coenzyme A. Okay, so this is clear. So odd chain fatty acid will produce propionyl coenzyme A and acetyl coenzyme A. Even chain fatty acid, on the other hand, will only produce acetyl coenzyme A, because once the even chain is broken, so C16 will go to 14, then 12, then 10, like that. If you keep on subtracting 2, you will get C4 and then C2. So all the carbons will be converted to acetyl coenzyme, and all these acetyl coenzyme can enter the TCA cycle. Clear? Moving on to the next question. Reverse cholesterol transport refers to the process by which? Option A, cholesterol is transported from the liver to peripheral tissue. Option B, cholesterol is transported from the peripheral tissues back to the liver. Option C, triacyl glycerides are transported from the intestine to the peripheral tissues. Option D, triacyl glycerides are transported from the peripheral tissue back to the liver. Reverse cholesterol transport refers to the process by which cholesterol is transported from the liver to the peripheral tissue. Cholesterol is transported from the peripheral tissues back to the liver. Option C, triglycerides are transported from the intestine to the peripheral tissue. Option D, triglycerides are transported from peripheral tissue back to the liver. You are saying option B, cholesterol is transported from the peripheral tissue back to the liver. That is the correct answer. So normally liver is the organ where the cholesterol is synthesized and then uh, transported across. But in reverse cholesterol transport, we have two main transporter proteins that basically transports the cholesterol back to the liver from the extrahepatic tissues. So you remember that HDL2, that was the final molecule, had the cholesterol esters and triacylglycerol. Triacylglycerol will be metabolized by the lipases, and the cholesterols that were present in HDL2, they will be transported back to the liver by a transporter called SRB1. Okay, so this is one transporter that is present in the liver membrane, and it will be it is a carrier of cholesterol from the extrahepatic tissue or from HDL to the liver, back to the liver. Second important transporter protein is ABC transporter protein, that is ABCA1. ABC is ATP binding cassette. So ABC transporter protein type A1. So this will basically transport the cholesterol to the pre-beta HDL, and then pre-beta HDL will become HDL2, and then this will be transported to the SRB1. But if you see in the intestinal membrane, we have this ABCA1 which transports the cholesterol from the intestine to the pre-beta HDL. So basically ABCA1 in the extrahepatic tissue, it is acting as a cholesterol transporter uh, to the liver. Okay, so ABCA1 is basically transporting the cholesterol from the extrahepatic tissue to the liver via HDL2. The ABCA1 that is present in the liver is transporting the cholesterol from the liver to the HDL2. So it is a back and forth mechanism. So the movement of cholesterol towards the liver is reverse cholesterol transport. Movement of cholesterol from the liver to the extrahepatic tissue, that's the cholesterol metabolic pathway, because liver synthesizes the cholesterol and then it is transported to different lipoproteins. Okay, that's clear, I hope. So moving to the next question, which apolipoprotein is required for the uptake of chylomicron remnants by the liver? Which apolipoprotein is required for the uptake of chylomicron remnants by the liver? Option A, ApoB100. Option B, ApoB48. Option C, ApoC2. And option D, ApoE. Which apolipoprotein is required for the uptake of chylomicron remnants by the liver? Option A, ApoB100. Option B, ApoB48. Option C, ApoC2. And option D, ApoE. So which apolipoprotein is actually required for the uptake of chylomicron remnants by the liver? Is it ApoB100, ApoB48, ApoC2 and ApoE? You're saying option B, ApoB48. Okay. So ApoB48 is the integral apoprotein for chylomicron we discussed, right? But here in this question, I'm asking which apolipoprotein is required for the uptake of chylomicron remnants. So the correct answer to this question is ApoE. So if you remember the chylomicron metabolism, we had this chylomicron, nascent chylomicron, which has the B48. So B48 is an integral protein for this chylomicron, and B48 is like the marker for chylomicron. Like if B48 is present, that means the molecule is chylomicron. It will mature with the help of other two apolipoproteins, ApoE and ApoC2. So ApoE and ApoC2, once they bind to this nascent chylomicron, it becomes a mature chylomicron. From this mature chylomicron, these triacylglycerides will be uh, metabolized by the lipoprotein lipase in the extrahepatic tissue, and they will get converted to fatty acids and glycerols, and the chylomicron will keep shrinking in size. So once the chylomicron is small enough where the cholesterol level is higher than the triacylglycerol, that chylomicron is called chylomicron remnant. Okay, and this chylomicron remnant is actually taken up by the liver. So my question was, which apoprotein is required for the uptake of chylomicron remnants by liver? So this chylomicron remnant will be taken up by liver by a receptor called LRP, that is LDL receptor related protein, and this receptor will bind to the ApoE that is bound to the chylomicron remnant. Okay, so we had chylomicron maturation by the binding of ApoE and ApoC2. Then ApoC2 will dissociate, and ApoE will remain bound to the chylomicron remnant. This chylomicron remnant, with along with this ApoE, now this ApoE can bind to this receptor LRP, that is LDL receptor related protein, and this chylomicron can be taken up by the liver where the cholesterol and fatty acids will be metabolized. Okay, so liver will have the cholesterol and fatty acids. So ApoE is the apoprotein that is responsible for the uptake of chylomicron remnant by the liver. Okay, so it's not the uptake of chylomicron, it's not the signature protein of chylomicron or integral apoprotein of chylomicron. It is the ligand of chylomicron remnant that binds to the receptor present in the liver for the uptake of chylomicron remnant. Okay, so it's called the remnant receptor or ApoE receptor or LRP, that is LDL receptor related protein. Is that clear? So ApoE is the apoprotein that binds to the LRP receptor, and chylomicron remnant is taken up by liver. Okay, moving to the next question. Which protein is essential for HDL function? Which protein is essential for HDL function? Option A, ApoA1. Option B, lecithin cholesterol acyl transferase. Option C, cholesterol ester transport protein. Option D, all of the above. Which protein is essential for HDL function? ApoA1, lecithin cholesterol acyl transferase or LCAT. Option C, cholesterol ester transport protein or CETP. And option D, all of the above. You are saying option D, all of the above. That is the correct answer. So HDL, so if you remember, pre-beta HDL has ApoA1 already attached to it. This ApoA1 pre-beta HDL, it will get converted to A1 with the get converted to HDL3 from the discoidal HDL by the help of the enzyme LCAT, that is lecithin cholesterol acyl transferase, which will convert the cholesterol into cholesterol esters. That cholesterol ester will then be exchanged with triacylglycerides from the different other lipoproteins like LDL and VLDL, and that exchanger protein or transport protein is called cholesterol ester transport protein, that will convert the HDL3 into HDL2, and then the cholesterol and cholesterol ester, they can be transported back to the liver by the receptor SRB1. Okay, so all the enzymes, all the proteins, ApoA1, lecithin cholesterol acyltransferase, and cholesterol ester transport protein, all three proteins are required for the HDL function. Okay, so any defect in any of these protein will lead to some metabolic disorder. Is that clear? Okay. Moving on to the next question. Entry of fatty acids into mitochondria for oxidation is inhibited by which factor? Entry of fatty acids into mitochondria for oxidation is inhibited by which factor? Option A, protein kinase A. Option B, AMP. Option C, epinephrine. Option D, glucagon. And option E, insulin. Entry of fatty acids into mitochondria for oxidation is inhibited by which factor? Option A, protein kinase A. Option B, AMP. Option C, epinephrine. Option D, glucagon. And option E, insulin. You are saying option E, insulin. That is the correct answer. So insulin basically inhibits the fatty acid oxidation. We'll see how. So this is the regulation of beta oxidation. So in case of high blood glucose, insulin is secreted and it signals towards the glucose uptake via glycolysis. So in higher level of insulin actually, higher level of insulin actually activates a phosphatase, and that phosphatase will dephosphorylate this ACC enzyme. Now, this enzyme is inactive when it is phosphorylated and it becomes active when it is dephosphorylated. So, high insulin will activate this phosphatase, and that will result to the activation of enzyme ACC. So high amount of insulin activating the phosphatase. That phosphatase will dephosphorylate the ACC enzyme, in turn activating it. That active ACC enzyme can now convert the acetyl coenzyme A into malonyl coenzyme A, which actually goes and inhibits the carnitine acyl transferase 1, that transfers the fatty acyl coenzyme A, that is the activated form of fatty acid, into the mitochondria. So fatty acids or fatty acyl coenzyme A, they cannot tra- travel through the mitochondrial membrane just like that. They are highly hydrophobic, and the membrane is hydrophobic in nature. Uh, so membrane is hydrophobic and hydrophilic, amphipathic in nature. So fatty acids or fatty acyl coenzyme, they cannot transport themselves on their own through the mitochondrial membrane. So they need a transfer protein, carnitine acyl transferase, and that phosphoprotein, we'll discuss about this. This is inhibited by malonyl coenzyme A. Okay, and it actually increases the fatty acid synthesis. So when high blood glucose is there, that will result in high amount of insulin, and high level of insulin will actually activate the fatty acid synthesis and inhibit the fatty acid breakdown. Just the opposite scenario, if we look at, that is low blood glucose. Now low blood glucose, glucagon is high. So insulin is high, that means glucose has to be broken down. There's a lot of glucose and glucose needs to be broken down by glycolysis. Low blood glucose means glucagon will be synthesized, and glucagon will trigger the trigger all the pathways that will go towards the glucose. Okay, so it will trigger gluconeogenesis and all the other pathways, glycogen breakdown and all. Okay, that will go towards the glucose. Now high glucagon also activates the protein kinase A. So we have, if you remember from the previous class, this protein kinase A was phosphorylating many enzymes in the glycogen breakdown metabolism, glycogenolysis, and that phosphorylation was actually activating the glycogenolytic uh, enzymes. Same concept is here. This protein kinase also phosphorylates the ACC, but the phosphorylated ACC is inactive. So in that case, when high levels of glucagon are present, the acetyl coenzyme A is not converted to malonyl coenzyme A. The acetyl coenzyme A actually, the high level of glucagon actually favors the beta fatty acid beta oxidation and lowers the fatty acid synthesis. Okay, so the question was, entry of fatty acid into mitochondria for oxidation is inhibited by which factor? So entry is inhibited by malonyl coenzyme A, and then malonyl coenzyme A is higher when high level of insulin is present. Okay, is this clear? Okay, moving on to the next question. Which of the following has the highest protein content among all lipoproteins? Highest protein content among all lipoproteins. Option A, chylomicrons. Option B, HDL. Option C, LDL. And option D, VLDL. Which of the following has the highest protein content among all lipoproteins? Option A, chylomicrons. Option B, HDL. Option C, LDL. And option D, VLDL. Which of the following has the highest protein content among all lipoproteins? Chylomicron, HDL, LDL and VLDL. So your answer is option B, that is HDL. That is the correct answer. So HDL has the highest protein content among all the other lipoproteins. So after HDL comes the LDL, VLDL, and then chylomicrons have the least amount of protein content. Chylomicron has almost 99% of lipids and only 1% of proteins. Okay, so HDL has the highest protein content among all lipoproteins. The next question is transport of fatty acids into mitochondria for beta oxidation depends on which molecule? The transport of fatty acid into mitochondria for beta oxidation depends on which molecule? Option A, coenzyme A. Option B, carnitine. Option C, biotin. Option D, NADPH. Transport of fatty acid into mitochondria for beta oxidation depends on which molecule? Option A, coenzyme A. Option B, carnitine. Option C, biotin. And option D, NADPH. Transport of fatty acid into the mitochondria for beta oxidation depends on which molecule? Option A, coenzyme A. Option B, carnitine. Option C, biotin. And option D, NADPH. You are saying option B, that is carnitine. That is the correct answer. So carnitine is the molecule that helps in the transport of fatty acid into the mitochondria. So I told you fatty acids can't uh, travel by themselves across the mitochondrial membrane. So it needs some assistance. So fatty acids are first activated to form fatty acyl coenzyme A, and then that fatty acid or the fatty acyl coenzyme A that will be transported across the mitochondrial membrane. So you know mitochondrial membrane is a double membrane. It has the outer mitochondrial membrane and the inner mitochondrial membrane, and between both the membranes there is a intermembrane space. So this fatty acid conversion of fatty acid or activation of fatty acid into fatty acyl coenzyme A that occurs in cytosol using the enzyme acyl coenzyme A synthetase. Okay, so acyl coenzyme A synthetase uh, will basically transfer the coenzyme A to the fatty acid. So acyl coenzyme A will be attached to the fatty acid. So instead of COOH, now you will have a coenzyme A group attached to the OH. So fatty acids becomes fatty acyl coenzyme A in the cytosol. That is the activated form that is required for beta oxidation. But now you know the enzymes of beta oxidation are present inside the mitochondria. So it has to reach the mitochondria. So fatty acyl coenzyme A has to travel through the outer and inner membrane of mitochondria to reach the mitochondrial matrix where the other enzymes of beta oxidation are present. Okay, so for that this fatty acyl coenzyme A will uh, be transported via carnitine. So there's the first enzyme that is involved is carnitine acyl transferase 1. So here you can see carnitine acyl transferase 1 is there. So carnitine acyl transferase 1 basically uh, gets so carnit basically attaches the carnitine to the fatty acyl coenzyme A. So the coenzyme A gets released, and instead of coenzyme A, now you have the carnitine. So you had this fatty acid to which the coenzyme A was added. Now this coenzyme A will be released, and in this place carnitine will be added. So this will be fatty acyl carnitine. So this was fatty acyl coenzyme A. Now instead of coenzyme A, you have fatty acyl carnitine. This fatty acyl carnitine that is converted by carnitine acyl transferase. This fatty acyl carnitine can be transported through the inner mitochondrial membrane by another protein called translocase. It's a transporter protein. This translocase has the ability to transport the fatty acyl carnitine to the other side of the membrane, that is the mitochondrial matrix. Now the fatty acyl carnitine has reached the mitochondrial matrix. But for beta oxidation, we want fatty acyl coenzyme A. So again, a coenzyme A has to be added to that fatty acid. So fatty acyl carnitine has to become fatty acyl coenzyme A for the beta oxidation to occur. So that transfer again is done by another acyl transferase, carnitine acyl transferase 2. So here we had carnitine acyl transferase 1 that was converting fatty acyl coenzyme A into fatty acyl carnitine by transferring the acyl carnitine to the fatty acid. Then this fatty acyl carnitine is carried to the other side of the mitochondrial matrix, to the other side of the mitochondrial membrane, or it is carried into the mitochondrial matrix by the transporter protein, that is translocase. And this translocase is carrying the fatty acyl carnitine and delivering it to the mitochondrial matrix. Here, this fatty acyl carnitine is again converted back to fatty acyl coenzyme A, and carnitine is released by the enzyme carnitine acyl transferase 2. So carnitine acyl transferase 1 was converting carnitine into fatty acyl carnitine. Carnitine acyl transferase 2 is converting fatty acyl carnitine to carnitine. Okay. Now this carnitine, the free carnitine, will be transported back to the outer membrane because we need to carry more fatty acyl coenzyme A from the cytosol to the mitochondrial matrix. So this carnitine that is transported back from the mitochondrial matrix to the outer membrane or cytosol, that transport again occurs via translocase, and this carnitine will then again bind to another fatty acyl coenzyme to become fatty acyl carnitine, and that fatty acyl carnitine can enter the mitochondrial matrix through translocase. Here, uh, along with the free carnitine, what we are getting? We are getting fatty acyl coenzyme A that can enter the beta oxidation. So all the enzymes of beta oxidation are present in the mitochondrial matrix. So once fatty acyl coenzyme A is present, it can all the enzymes can get activated, and they will be converting the fatty acyl coenzyme A into acetyl coenzyme A that can enter the TCA cycle. Okay, so fatty acid getting converted into its activated form, that is fatty acyl coenzyme A. Fatty acyl coenzyme A now has to reach the mitochondrial matrix, which it cannot do on its own. So carnitine is the molecule that will act as a transporter. So for that, carnitine will get attached to the fatty acid at the acyl by the carnitine acyl transferase 1, and we'll get fatty acyl carnitine. That fatty acyl carnitine will be transported via translocase protein or transporter protein. That translocation of fatty acyl carnitine to the mitochondrial matrix will occur, and then another acyl transferase, that is carnitine acyl transferase 2, will convert the fatty acyl carnitine to fatty acyl coenzyme A, and free carnitine will be released. That will be transported back to the outer membrane through the same transporter protein, translocase, and this carnitine can bind to a new molecule of fatty acyl coenzyme A. So all the fatty acids have to be converted to fatty acyl coenzyme A in order to reach the mitochondrial matrix. Is this clear? If you have any doubt in this, please let me know. Okay. Okay. Moving to the next question. This is actually the last question of my slide. So, which enzyme catalyzes the first step in beta oxidation of fatty acids? Which enzyme catalyzes the first step in beta oxidation of fatty acid? Option A, acyl coenzyme A dehydrogenase. Option B, thiolase. Option C, enoyl coenzyme A hydratase. And option D, carnitine acyl transferase. Which enzyme catalyzes the first step in beta oxidation of fatty acid? The first step of beta oxidation. Option A, acyl coenzyme A dehydrogenase. Option B, thiolase. Option C, enoyl coenzyme A hydratase. And option D, carnitine acyl transferase. Which enzyme catalyzes the first step in beta oxidation of fatty acid? Acyl coenzyme A dehydrogenase. Option B, thiolase. Option C, enoyl coenzyme A hydratase. And option D, carnitine acyl transferase. You are saying option A, that is acyl coenzyme A dehydrogenase. That is the correct answer. So this is the beta oxidation of fatty acid. So here the example is of palmitoyl coenzyme A, which is a 16 carbon fatty acid. So beta oxidation obviously is the breakdown of the fatty acid at the beta carbon. So the naming is done such that it starts from the functional group. So fatty acid has the COH group, that is the functional group, and later it is activated to form fatty acyl coenzyme A, as we saw in the previous slide. That fatty acyl coenzyme A, uh, that so this acyl group basically is the main functional group. So the first carbon attached to this acyl group will be the alpha carbon. After that we'll have beta, gamma, and so on. So in beta oxidation, the breakdown occurs from the between the like after the beta carbon. Okay, so this palmitoyl coenzyme A, that is 16 carbon fatty acid, the first step in beta oxidation is carried out by the enzyme acyl coenzyme A dehydrogenase, which is FAD containing dehydrogenase. So I told you in the previous classes also, generally when the enzyme dehydrogenase is present, it usually has a electron carrier, either FAD or NAD. So it's a reduction reaction. So here, acyl coenzyme A dehydrogenase, which is a FAD containing enzyme. So FAD will be converted to FADH2, which will carry the electrons, and in return, the two hydrogens will be removed. So this is a dehydrogenation step. So you see from this beta and alpha carbon, one hydrogens are removed. So instead of CH2, now it is CH, and instead of this CH2, this is also CH. So, and one double bond is introduced between the alpha and the beta carbon. Okay, so that is the role of acyl coenzyme A dehydrogenase, that will basically do the dehydrogenation from the alpha and beta carbon, removing one hydrogen each from each carbon, and introducing a double bond between the alpha and the beta carbon. Okay, then comes the next enzyme, enoyl coenzyme A hydratase. So enoyl coenzyme A hydratase basically introduces one H2O molecule to this beta carbon. So you see it was CH. So one OH is added to the beta carbon and hydrogen is added to the alpha carbon, breaking the double bond that was present here. So this double bond is a trans double bond. You can see both the hydrogen are in opposite directions. It's a trans delta 2 enoyl coenzyme A. Trans enoyl coenzyme A will then be converted to L beta hydroxy acyl coenzyme A. So beta carbon has a hydroxy group now. So it's called the beta hydroxy acyl coenzyme A. So we started with the fatty acyl coenzyme A, that was palmitoyl coenzyme A in this example. This will become trans enoyl coenzyme A because there's a enol group. Double bond is generally referred to as ene, and this double bond is trans in nature because both the hydrogen, as you can see, are in opposite direction. They are not in the same direction of the double bond. They are in the opposite direction of the double bond. So it's a trans enoyl coenzyme A. Then enoyl coenzyme A hydratase, which will introduce a H2O or a water molecule in and break down the double bond. This will lead to the formation of beta hydroxyacyl coenzyme A, because the beta carbon now will have the OH group, the hydroxy group, and the double bond is now broken. Now the presence of this hydroxy group makes it uh, easier to be dehydrogenated. So again, a dehydrogenation reaction occurs. So first one was dehydrogenation reaction, then a hydration reaction, again a dehydrogenation reaction occurs. This time this dehydrogenase has NAD molecule attached to it. So in the first dehydrogenation step, we had a FAD cofactor that was converted to FADH2 carrying the electrons. In this case of dehydrogenation, we have NAD molecule that will carry the that will get converted to NADH, and it will carry the electron. This NADH and FADH2 both can enter the electron transport chain, producing ATP. So the second dehydrogenase is called beta hydroxyacyl coenzyme A dehydrogenase. So whatever the substrate is, beta hydroxyacyl coenzyme A is the substrate. So dehydrogenation of this substrate is occurring. So beta hydroxyacyl coenzyme A dehydrogenase. Okay. Now what does this dehydrogenase do? It removes the hydrogen from the beta carbon. So now removal of both the hydrogen from the beta carbon leaves with a ketone group. C double bond O, ketone group is there. See, the beta carbon now is in a ketone group, and this is called beta ketoacyl coenzyme A. Okay, this compound is called beta ketoacyl coenzyme A. Now this ketone group will now be easier for a coenzyme A transfer. So what will happen? One acetyl transferase, basically acyl coenzyme acetyl transferase, or ACAT, which is also called thiolase. So this this enzyme will transfer the coenzyme A to the beta carbon, and the 2 carbon will be eliminated as acetyl coenzyme A from the fatty acid. Okay, so we had the fatty acid which was 16 carbon length. This fatty acid was activated to form the fatty acyl coenzyme A. So from this fatty acyl coenzyme A, first dehydrogenation step left us with a double bond between the alpha and the beta carbon, uh, and the product was called trans delta 2 enoyl coenzyme A. Then a hydration steps occurs with the enoyl coenzyme A hydratase, where the H2O is introduced at the beta carbon and the double bond is removed. This leads to the formation of beta acyl coenzyme A, because there's a hydroxy group, OH group, on the beta carbon. Then this beta carbon becomes more susceptible to dehydrogenation. This dehydrogenation step is carried out by beta hydroxyacyl coenzyme dehydrogenase, which is a NAD containing enzyme, and the NAD will be converted to NADH, while beta hydroxyacyl coenzyme A will be dehydrogenated. The dehydrogenation leads to the ketone group formation at the beta carbon, and it is called beta ketoacyl coenzyme A, and that ketone group will be then attacked by the coenzyme A, and this coenzyme A transfer will be carried out by acyl coenzyme A acetyl transferase. Acyl coenzyme A acetyl transferase, that is ACAT, and it is also called thiolase. So because a thiol bond will be forming, so SH is the thiol group, and now there will be an elimination of the hydrogen, and then carbon will be attached to this thiol group of the coenzyme A. And this bond between the alpha and the beta carbon will be broken down, and the alpha carbon and the acyl coenzyme A will get removed, leading us with the molecule called acetyl coenzyme A. And this 14 carbon, remaining 14 carbon fatty acyl coenzyme A can again enter another round of beta oxidation, where again two carbons will be removed and we will be left with C12 uh, acyl coenzyme A, fatty acyl coenzyme A. Then from C12 we will have C10, then C8, and so on, until all the carbons are broken, like all the fatty acids are broken down into 2 carbon acetyl coenzyme A. Okay, so 16 carbon fatty acyl coenzyme A can go through I think 7 cycles, and we'll get total 8 acetyl coenzyme A. Okay, and all these acetyl coenzyme A, you know where it can enter. It can enter the TCA cycle, which ultimately will lead to uh, the ATP production. Okay, is this clear to you? Do you have any doubt in any of the questions discussed or any of the topics? Anything, if you have any doubt which is not discussed by me, you can ask me. Like for week 5, all the topics of lipoprotein metabolism and fatty acid catabolism. If you have any doubt in any of the topic, you can let me know. If I missed any topic to include in the questions, I can try to explain. If you have any doubt, let me know. So you have seen, so I have discussed about the lipoprotein metabolism, where different lipoproteins like chylomicrons, VLDL, LDL, and HDL. We have discussed the metabolism of all these lipoproteins. So all these lipoproteins are associated with different apoproteins, and some of the apoproteins are integral apoproteins, which are like signature markers of those lipoproteins, and some of the apoproteins are peripheral apoproteins, which binds to them when it's required, and it gets detached when it's not required. For the maturation, mostly ApoE and ApoC2 are attached to the lipoproteins for the maturation of those lipoproteins. So nascent chylomicron converts into mature chylomicron, and then nascent VLDL matures into VLDL. So, and all these lipoproteins are carrying the triacyl glycerides and cholesterol. So highest amount of triacyl glyceride is carried by chylomicrons, and highest amount of cholesterol is carried by HDL. Highest amount of protein is present in HDL, and highest amount of lipid is present in chylomicron. So these are the basic MCQs that are like very common in these topics. Then we saw the different uh, so there are multiple disorders associated to the lipoprotein metabolism that are discussed in the lecture, so you can go through the tables. I have not included many questions related to the disorder. There are disorders related to the beta oxidation or the fatty acid catabolism pathways also, so I would request you to go through the disorders as well. And after lipoprotein metabolism, we move to the fatty acid catabolism, where the fatty acids are broken down by different oxidation pathways. So we discussed about beta oxidation in detail. Okay. So do you have any doubt? Okay, welcome. So, if you don't have any doubt, then I think I can end this session now. So, thank you for joining. I'll end the session here. You can access the video of this class through the YouTube link shared, and I'll share the slides as well, so you can refer to it anytime. Okay. So, thank you so much. I'll end this call now. Bye bye.